So maybe we'll have to do.
Yeah.
Yeah, we're happy to talk. We have seen stuff on bunch of others.
Serious. I have to go over there.
Where he is.
Yeah, we're gonna get started pretty soon here. Okay, it's my great pleasure to introduce James DiCarlo. He came to us from Johns Hopkins University, where he was working, in addition to working on all his other undergraduate work, he was doing research with an old friend of mine, Jeff Bouquet. And during that period of time, from 2006 to 2010, he participated in synthesizing chromosomes towards synthesizing a whole yeast genome, the first eukaryotic synthesis, genome synthesis. And he participated in 2 iGEM, International Genetically Engineered Machines competitions. His team winning a bronze in 2008 and a gold in 2009. And so when he came to the PhD program at Boston University around 2010 and showed an interest in our group, I was really torn. He seemed like kind of marginally relevant to our laboratory. But we let him in, and there's absolutely no regrets. It's terrific. It's been absolutely terrific. He's been the core of our yeast projects from day one. I knew he was a ringer coming from Jeff's lab, and he certainly was, and went well beyond yeast, as you'll hear, in its implications to gene drives. But again and again, funding agencies would say, we basically just want to fund James' work, that's it. And so he really had a big impact on our funding as well from NSF and other groups. So, take it away.
Thanks, Chris. Thank you so much. Thank you everyone for coming, especially people that came from far away. So the title of my defense talk today is Engineering Yeast Genomes and Populations. So it's important to start out with defining exactly what a genome is. So a genome is the complete set of DNA that is within an organism. And the DNA is what makes up your genes. And these genes are organized into chromosomes. And they're found within the nucleus of the cell. And what the genome is basically a blueprint for the organism. And you can kind of think of it as like a hard drive in your computer, where all the information is stored and encoded into the hard drive. And the genome is like the hard drive of the organism. So information on hard drives can be encoded into different programs, such as like Angry Birds or TurboTax, 2 very fun programs that everyone's familiar with, I'm sure. And similarly, information in genomes can be encoded into different organisms, such as wolves or platypuses, or anything really, any living organism pretty much. But what's cool about organizing information into different programs is that a computer programmer can easily take a program like Angry Birds and modify it and turn it into like a spin-off program like Angry Street, which is a parody program of Angry Birds. And people have been doing the same thing for organisms for thousands of years. And if you take the wolf, for example, people have turned that into something more familiar to the domestic dog. So if you compare those 2 modified programs, the methods that they employed to make these programs different are pretty, they're also different. So to edit the Angry Birds into Angry Street, what computer programmers did is edit the code of the program. But what people did for wolves into domestic dogs was selectively breed the organism. And there's a big difference between those 2 things, is that the time scale for editing the code of Angry Birds was much shorter than the timescale of editing the wolf. And that's because of how long it takes to selectively breed an organism. So the solution to get this a lot shorter is to selectively edit the code of the organism. And editing the code and precisely modifying the genome of an organism or computer code allows for much quicker and predictable changes to be made. It allows you to study components, the function of the components that are already in place in the organism, and it also allows you to design new functionalities into the organism. So, as George mentioned, the organism that I focused the majority of my thesis on was budding yeast, and I'm sure most people have heard of budding yeast or baker's yeast. It's been a crucial organism for food and beverage production for over 8,000 years. Since the 1970s, it's been an important organism for industrial bioproduction. For pharmaceuticals, people have made artemisinic acid, which is a precursor to an antimalarial drug. People make various epitope-based vaccines in yeast, such as Recombavax, which is a hepatitis B vaccine. Many different vitamins are synthesized in yeast industrially, as well as recently a group, John Buber's group, synthesized one of the opioid biosynthetic pathways, so it could revolutionize some of how those pharmaceuticals are made. Also, people have made bioplastics in yeast. Since the 1960s or so, or a little earlier, people have used yeast as a model for eukaryotic biology. About 23% of yeast genes have very high homology to human genes. So a lot of the pathways in yeast, especially highly conserved pathways like DNA replication and repair, can be studied in yeast, and the implications can be carried through into humans. And a study recently took 76 essential genes from yeast and replaced them completely with the human homologue, and the yeast function just very well. And lastly, in the past 20 years or so, people have started to use yeast as a chassis or an organism to hold large pieces of DNA. In 2009, I think it was, the Venture Group synthesized an entirely new bacterial genome in yeast. So that's another more recent use that yeast has been used for. So currently, to modify yeast genomes, you can do two things. You can modify the actual genome, which is composed of linear chromosomes, or you can modify plasmids. And plasmids are circular pieces of DNA, and they're kind of like USB drives or like floppy disks. They're pretty small, and they require selection as well, either in the form of an antibiotic gene or an oxytrophic marker. And you can see like a little diagram below with the linear chromosomes versus the plasmid that's circulating in the nucleus. An important thing to note about genomic changes versus plasmids is that genomic changes are much more stably expressed, and they're also more predictably expressed. So if you compare, this is a chart of two different fluorescent proteins expressed under different promoters, and if you look at the genome expression profiles versus the plasmid expression profiles, the plasmid ones are highly variable and kind of a mess in some ways. But the genome expression profiles are much more regular and predictable. And this is important for engineering different pathways, or if you want to precisely engineer an organism to have very predictable expression profiles. So that's one of the big advantages of engineering things on the genome. So currently, how we modify yeast genomes is we take advantage of this very powerful system that's found in pretty much all organisms, but it happens at different rates. And this system is called homologous recombination. And the basic way that this happens is it relies on pieces of DNA that are the same between the introduced piece of DNA and the host genome. So in the first step, you take the host genome and you have these homology arms, which are the same as the introduced foreign piece of DNA. This is the same sequence. So what happens is a huge cascade of proteins recognizes that these two pieces of DNA are exactly the same, and they catalyze the homologous recombination reaction in which the foreign piece of DNA is recombined into the host genome because of these similar pieces of DNA here. And this is like designated by 2 X's often in genetics. And then in the second step, this is very simplified. In the second step, the DNA is integrated into the host genome. And this is carried out by a ton of different proteins. So as a little, just to get everyone's frame of reference, if you look at current rates of homologous recombination in yeast, using short homology arms, about 0.001% of the cells that you put this foreign piece of DNA into will actually accept the change and integrate the DNA. When you use longer homology arms, when these pieces become even bigger, you can up it by about 100-fold. So that's kind of like where we are at the beginning of my thesis. So the goal of my thesis was to make a technique to modify genomes with high efficiency using short pieces of DNA, because short fragments can add up to large changes if you do the process over and over again. The second goal was to explore factors that stimulate the ability to engineer genomes, specifically looking at reprogrammable endonucleases that cut DNA. These are like enzymes that break the code in the DNA and allow the code to be easier to be amended. And lastly, to test the use of genetic devices that spread changes across populations. These are kind of like self-spreading code, and I'll describe all these a little more. So an overview of the talk today, I'm going to talk about a technique I designed for incorporating oligonucleotides, which are small pieces of DNA. Look at the use of reprogrammable endonucleases, and then also investigate the feasibility of gene drives and using these self-spreading code, they're also called selfish genes, to spread traits across yeast genome populations, or yeast populations. So the first technique I'm going to talk about involves those short pieces of DNA called oligonucleotides. And we came up with a name. My collaborator who worked with on this project, Andrew Conley, actually came up with an acronym for yeast oligonucleotide mediated genome engineering, or YOGI, as a little affectionate acronym. So why would we use oligonucleotides? So just as a refresher, oligos are short DNA molecules. They're very cheaply synthesized, which is really nice because you can make a lot of them. They're the appropriate size to modify different regulatory regions or protein coding regions, so they're of a usable size that you could actually use them for genome engineering. And the changes that are made with oligonucleotides can add up. So if you have these like linear chromosomes here and you incorporate oligos, over time, if you do this in many times, you can end up with chromosomes that look very different than when you started because the changes are additive. So the overview of what I'm going to mention about Yogi, I'm going to talk about the reporter systems I used, as well as the different endogenous pathways that I manipulated. And I'm going to lastly mention some of the cycling I did so that the additive changes could be studied. So starting off, I took a wild-type yeast cell and I transformed in an oligo. And transformation is just the process where you put a piece of foreign DNA into a cell. So I transformed in an oligonucleotide, and I looked for the change that was made. And the way I assayed how well the change was made is I measured the recombination frequency, which is a ratio. And it's the ratio of number of cells that incorporated the oligo versus the total number of surviving cells. And I looked at 5 different markers. So these are 5 different genes that I incorporated oligonucleotides into. 2 of them were broken, and I fixed them with an oligo. And 3 of them were already working, and I broke them with an oligo. And these could all be assayed very well. So the 2 endogenous pathways that I manipulated in yeast were the mismatch repair pathway, which deals with heteroduplexed, partially complementary DNA, which is important because when I introduce an oligo, there's going to be a region that I want to change, and this region will be not exactly the same as the one in the genome. And so this pathway deals with DNA heteroduplex DNA recognition and excision. And the second pathway is the homologous recombination pathway that I mentioned before. And it's present across all domains, and it's very important for fixing broken DNA and broken genomes. So this is a little cartoon of how a mismatch repair works. So there are three main players. So there are the first complex, the MSH2, MSH6 complex. The second is the MLH1, TMS1 complex, and the third is XO1, which is a DNA cutting enzyme. So when you have mismatched DNA, it's represented kind of as like a bubble. So these regions are complementary to each other, but these regions are not. So they don't participate in traditional base pairing. So you have it's conceptualized as a bubble. So what happens is that the MSH2, MSH6 complex recognizes this little bubble and recruits the MLH1, PMS1 complex. This then in turn recruits the exo and nuclease, which then cleaves away this bubble. And this can then be repaired by other proteins in the system. So because the changes that we were introducing to these genome would create a bubble like this, we wanted to see if we knocked out these particular enzymes, if we'd have an effect on the incorporation frequency of oligos. And what we found is that when we knocked out the MLH1 gene in 3 different strains, so let me first say that we worked with 3 different strains. This is all this work's also done with Andrew Conley, who was a visitor in our lab. So the 3 strains we worked with were the VL648 strain, which is a lab strain that was reported to have high recombination frequency already. We worked with the SenPK strain, which is a more industrial strain, but it's also a good lab strain. And then we worked with this industrial yeast strain, this VGT strain that was originally identified as a brewery contaminant. So it's very robust and it's really cool in industrial settings, but it's a little harder to engineer. So we picked those 3 strains and we knocked out MLH1. And what we found is that when we knocked out MLH1 across all the different areas that we incorporated oligos into, so in these 2 strains we had 3 different targets, 3 genes that we incorporated oligos into. In this strain we only just incorporated into 1 gene. But we found that the incorporation frequency ranged from anywhere from 3.8 to 11-fold increase when we knocked out those genes. When we knocked out the MSH2 gene, we found that the incorporation frequency was a little less, anywhere from 3 to 9-fold increase. And when we did the double knockout of both MSH1 and MSH2 in these 2 strains, the incorporation frequency went a little bit higher, up to about 13. However, these 2 strains had a growth phenotype that was much slower, so we decided to just stick with the MLH1 strain going forward because this strain gave the highest fold increase in oligonucleotide recombination. And we stuck with this knockout going forward for all 3 strains. We then wanted to manipulate the homologous recombination pathway that I mentioned before, and this is just a refresher. DNA with homology arms can be incorporated into genomes via a bunch of different proteins. And this pathway is quite complex. And here's just one branch of the pathway involved with single-stranded DNA, in which multiple proteins are involved in the recombination. But we actually, a few papers had already reported that RAD51 and RAD54 were important in the repair using single-stranded DNA. So we decided to just focus on those 2 proteins. We also looked at a particular mutant of RAD51. There's a point mutant that also had an increased ability to recombine single-stranded DNA. So we focused on just RAD51 and RAD54. And if you compare, so when we just incorporated them singly into all 3 strains, the incorporation frequency for just RAD51 versus the RAD51 single mutant was 27 versus 80-fold, so it's about 2-fold more, a little more than 2-fold more with the mutant RAD51 in the VL6 strain. So we took this mutant RAD51 and incorporated it into the other 2 strains. And what we found is a incorporation frequency ranging from anywhere from 5.8 at the lowest up to 26 in these 2 strains. But it was much higher in the VL6 strain actually. And we also incorporated the RAD54 gene singly into this SenPK strain, and also saw a similar increase in oligonucleotide recombination. When we did the double overexpression of both of those genes, we found is a oligo recombination frequency ranging incorporation fold by anywhere from 20 to 500 fold. And that corresponded to recombination frequencies anywhere from 0.2 to 2%, depending on the locus in which we all incorporated oligos. So that was really high. That was much higher than anyone had reported before. So the types of changes that can be made with these oligos, we took the best strain that would recombine oligos, and what we did is we tested different types of mutations that could be made. And mismatches, which are pieces of DNA which don't have the same base that you're incorporating, and did much better for incorporation than deletions, and that's where, or insertions. So in deletions, you actually remove segments of DNA, and in insertions, you insert pieces of DNA. And it varied depending on how many base pairs we actually incorporated. So with 1 to 8 mismatches, there was a decreasing homologous recombination frequency. But with deletions and insertions, it stayed around the same, and it was much lower than single base mismatches. So multiplex modifications could be made using this method if you cycle the changes, if you cycle the process. So starting with the cells that, the most recombinogenic cells, if you incorporate oligos in a cyclical way, where we start with the cells in this state here, prepare the cells and transform in DNA, and then transform the cells with DNA, and then we do this cyclically, so we start at 1, go to 2, and go to 3, and do this for 3 times. We wanted to measure to see if we could increase the amount of recombination in a cumulative way. So after the 3 steps, we would assay the recombination frequency, and then we do this cycle 3 times, and we'd assay the cumulative recombination frequency at the end of each cycle. And we picked 2 different alleles, or 2 different genes that we wanted to target. And what we found is that after each cycle, the cumulative recombination frequency increased. And so at the end of cycle 3, the recombination frequency at each locus was anywhere from 0.5 to 0.7 for these 2 genes that we targeted. And in the combinatorial setup, where we assayed the mutation frequency at both loci in one cell, the frequency was about 0.005. So this shows that you can complete this method in a cyclical way and accumulate changes over time. So you can start with something where you have a bunch of wild-type chromosomes, incorporate oligos, and over time you'll end up with a genome that is much different than you started. So as a little summary for the Yogi process that we engineered, the potential library size for this method was anywhere from 100 to 10 to the 5 recombinants per locus per cycle. And that just depends on the locus that you target as well as the strain that you use. And that's just inherent variation in both recombination efficiency at each different locus as well as the ability for different strains to incorporate oligos. And the final recombination frequency ranged from anywhere from 0.2 to 2% for all strains of loci. And if you compare the original chart we had, where we had 0.001% incorporation frequency with short homology arms and 0.1% recombination frequency for longer arms, when you have Yogi and short homology arms, you get a frequency of anywhere about 1%. So we're doing much better when you actually employ Yogi. So the second step that I took in my thesis was to look at reprogrammable endonucleases. And these are super exciting. And these come from the CRISPR-Cas system. And I'll describe all these things in a little more detail. So the reason we looked at endonucleases is because these are DNA scissors that cut within DNA strands. So they're often represented as like Pac-Man that cut, and they don't really eat DNA, but they cut it and then make a break in the DNA. And DNA that is broken has a much higher recombination efficiency. So in yeast, if you break the genome and incorporate a piece of DNA, the recombination frequency can be increased by about 4,000 fold. So that's much higher. So this is a little schematic of what happens. You have your introduced DNA, and you have these homology arms that we mentioned before, and then you have your genome. When you cut the DNA using these Pac-Man or DNA scissors, as they're often called, the introduced DNA can be integrated via homologous recombination, and this is stimulated because of the break. So this is a case in which you do have a piece of DNA to introduce. And then the DNA is recombined into the genome using homologous recombination. There's also a scenario in which you don't have this introduced DNA, and what happens is you get mutagenic repair. So the area in the genome is mutagenized. The cell wants to repair this cut somehow, so it just usually either anneals the ends or puts in some bases, basically. So those are the different pathways that we looked at. So the types of the type of endonucleases that we looked at for this study were from the type 2 CRISPR-Cas system from Pyogenes streptococcus. And basically this is a bacterial immune system. So Pyogenes streptococcus is a bacteria, and it employs these cluster regulatory interspace short palindromic repeats, and that's what CRISPR stands for. It employs this system to protect itself from invading nucleic acids, such as parasitic genetic elements like bacteriophages, which are viruses. So this is basically an immune system for bacteria. So we took this system, we took several components of the system, and we put them in yeast, basically, and see how well they work. But here's a little diagram of the native system in Streptomyces pyogenes, Streptococcus. So in that particular organism, CRISPR is a 3-component system where you have a CRISPR RNA, and this associates with a region in the genome, a transacting RNA, which coordinates with both the CRISPR RNA and the Cas9 protein, and the Cas9 endonuclease. So all of these components come together to form a functional DNA cutting enzyme. So you need 3 components for the native system. In 2012, the Duodna group came up with a new RNA that works just as well as the 2 previous RNAs. And basically what they did is they fused the CRISPR RNA to the tracer RNA in a particular way, so that all you need is a synthetic guide RNA and the Cas9 protein to form a functional endonuclease. So they took a 3-component system and made it a 2-component system, which makes it a lot easier to use. So here's a close-up of Cas9 in association with the guide RNA. So this is the genomic locus that you want to cut. This is the protospacer associated motif, and this is a required region for Cas9 cleavage. However, this region here can be completely recoded. So this can be any region in the genome as long as it's next to an NGG sequence. So in this case it's a TGG, but this can be any sequence as long as it's near an NGG. So what makes it amazing is that you can design a DNA cutting enzyme to any locus in the genome as long as it has an NGG sequence. So this has revolutionized the genome engineering field for that reason. So what I wanted to do in yeast was validate that Cas9 was being expressed and actually cleaving DNA. And so I looked at targeted mutagenesis. And so that was the scenario in which you didn't supply a foreign piece of DNA. Then I looked at the recombination frequency with foreign DNA, and then I optimized the technique and applied it to a particular proof of concept. So this is just another diagram of Cas9 in association with the locus, and you can see the cutting happening here next to the NGG. So I took Cas9, expressed it with a particular promoter. I took the guide RNA, expressed it with a SNO RNA promoter here. And then I took 2 different targets. I took the at the CAN1 gene, so I called them CAN1Y and CAN1Z. So they're just 2 different targets of the same gene. And they have different PAMs and they target different sequences. So what I did is I first looked at mutagenesis. So this was the scenario in which I didn't supply a foreign piece of DNA. So you should get mutagenic repair. So we should see an increase in mutation frequency at the CAN1 gene. So I looked at 2 different genes. I targeted the CAN1 gene, and then I looked at I monitored the mutation frequency of the LIPE1 gene, which is another negatively selectable marker. So I could monitor the mutation frequency of both the targeted and a non-targeted gene. And the reason I chose a non-targeted gene as well, it was just to make sure that the targeting was specific to the CAN1 gene. So within cells, when I express Cas9, or when I just expressed the two different gRNAs, the mutation frequency did not increase at the CAN1 gene. However, when I expressed both of those components together, we saw a much higher mutation frequency that was increased by over 1,000-fold, and that depended on the site that we targeted. So that was a great indication that Cas9 was actually cutting. And you also can see that the LIFE1 non-targeted gene, the mutation frequency stayed about the same. So that meant that the cutting was actually specific, or mostly specific. We then sequenced strains that emerged from this study, and we found that the mutations that were incorporated into the genome were very close to the PAM. So that was another indication that Cas9 was cleaving correctly at the specified locus. You can see you have a lot of deletions, and you have a few insertions here. And these were the two different PAMs for the two different targets of the CAN1 gene. So that was nice validation too. So then I wanted to see if we could take this system and use it to stimulate DNA recombination. So what I did is I took a gRNA plasmid, so I expressed that gRNA component on a plasmid, which I could select for. And then I incorporated either double-stranded oligos or double-stranded larger pieces of DNA. So these are up to 2,000 base pairs. So oligos are anywhere from like 2 base pairs, I guess, to like about 200. So these are much larger than the oligos. So I then transformed these into yeast, and I looked at the recombination frequency of either oligos or the double-stranded cassette. So without the gRNA, the recombination frequency of both the oligo and the cassette were very low. However, when I incorporated the gRNA into the plasmid and looked at the incorporation of both the oligo and of the larger piece of DNA, the recombination frequency was almost 100%. So that was quite impressive, because this means that you could incorporate changes onto the yeast genome without providing selection at the locus of incorporation. So that was pretty exciting. So what I wanted to do is then apply this system to a proof of concept experiment. And I took a phenotype in yeast that could be easily assayed, and that was thermotolerance. So several gene knockouts actually in yeast have been shown to benefit stress response to high heat. So actually when you knock out these 3 genes in yeast singly, the knockout actually confers thermotolerance. So they can actually survive higher heat stress, which is pretty interesting. And the thought is that it kind of preconditions the cell to a stress responsive state. These are all involved in like very complex pathways. And then another gene I took was a ubiquitin gene. So this gene deals with marking damaged proteins for degradation. And this particular gene, when it lacks a phosphorylation site, it confers a thermotolerant phenotype. And it's thought in the same way that it's kind of preparing the cell for a stress-responsive state. So I wanted to look to see if I could make changes to these 4 genes in a multiplex fashion. And that way I could look at how these genes affect each other, and if they confer an even greater thermotolerant resistance phenotype. So I took the 4 genes, and I designed gRNA plasmids to target each of the genes, and I also designed 4 different donor oligos that would incorporate either stop codons, which would knock out the 3 genes that need to be knocked out, or a mutation that would remove the phosphorylation site for this particular gene. And I co-transformed all of these as a pool into yeast. And I did a similar cycle that I did before with Yogi, where I transformed the cells, I selected for the gRNA plasmid, and I assayed the recombination frequency at the loci that I was looking at. And then I cured the population of the plasmid, and I did this 2 times. And what I found is that after 1 cycle, the vast majority of the cells had 1 or more change. And then after 2 cycles, about 40% of the cells were double mutants. And I was actually able to identify a triple mutant as well. So doing serial dilutions of these strains that I identified, so starting with, so I was able to get, I think, 9 different strains out of the process. They're either single, double, and then 1 triple mutant. So what I did is I took the cells, and I did a serial dilution, which means you start with very dense cells up here, and you do 10-fold dilutions down the plate. So just starting out with no incubation, you can see the cells kind of grow, and they have the same kind of density, where they're the same amount of cells in each different well. Then I did a heat shock at 55 degrees for 2 hours, and I plated these cells in the same way. And what you can see is that in the single mutants, as reported in the literature, a few of the different single mutants have, this one seems to have a much better fitness than the wild type cell. And then if you look at the double mutants, it looks like this one here, this double mutant SCH and TFS1, have a higher fitness, as well as this TFS1 UBC mutant here. They both seem to have a higher fitness than the wild type. So it was kind of cool. It shows that using CRISPR-Cas to make multiple changes in yeast, you can study epistasis between genes. So that's what so in studying the interaction between genes. So as a summary for the yeast CRISPR-Cas experiments, I optimized the CRISPR-Cas system for genome engineering in yeast. And with this system, I found that you could generate libraries as high as 10 to the 5th with genomic recombinance. And I also applied this technique to thermotolerance. And looking at the chart that we had before, where I added Yogi, using CRISPR, you can get recombination frequencies of upwards of 100%. So you can't get better than that, pretty much. But we always aim to. So then the last part of my talk, I want to mention some of the work I did on gene drives, which are selfish genes to modify yeast populations. So when we want to introduce an allele or a gene to a population, the chance of passing that gene on is only about 50% unless there's selection for that particular gene. And in this case, if you take these two yeasts, for example, I first want to say that yeast, for the most part, reproduce asexually, so they just divide. However, you can get them to mate sexually as well. So in